All right, it seems like we have some critical mass here, so I think we can go ahead and get started. Thank you all for joining today's Knowledge Portal Network webinar. Today we have an exciting talk from Emily Griffin from the Gaulton Lab. But before we get started, I'm going to walk through some events and upcoming webinars that you can attend to learn a little bit more about, us and the work that we do. So the first one that I just want to invite you all to is our June 29th virtual roundtable with the NIH's All of Us research program. This will be from 1 to 2 p.m. Eastern. And please feel free to scan the QR code here or put in this link to register yourself for this roundtable. You can hear from our researchers using All of Us data, Satoshi and for Julie, both from the Broad Institute and members of our group. And you might want to attend this event because this gives you a great opportunity to see how to use the All of Us data to study diabetes at scale. And also use this as a way to explore real world evidence, genomics, and longitudinal data. And this will be a great place for you to share your perspectives, in addition to hearing from perspectives from Satoshi and Julie. So again, we invite you to join us on Monday. Again, please feel free to scan the code or put in the link into your website to see the registration information for this session. And if you have any questions or concerns about this and you need help with registration, please let me know. Next, we are going to be attending the Intelligent Systems for Molecular Biology conference in Washington, D.C., July 12th through 16th. Julie Jurgens from our team will be presenting a short talk, The Common Fund Data Ecosystem, CFDE, Incubator for Pan-NIH Resources, Enhancing Interoperability. And this talk will be during the Bosch track on July 15th from 2:40 to 3 p.m. She'll also give a presentation as part of the poster session as well. And we'll talk about our CFDE resource, the Knowledge Center. On the right here, I just wanted to highlight some other programs from the Common Fund Data Ecosystem who will also be participating in this conference. So if you're going to be attending, please keep an eye out for Julie and also for the presentations from our other ecosystem attendees. So we really look forward to that coming in the next few weeks. We also have the Common Fund Data Ecosystem public webinar series. And so tomorrow's session will be from 1 to 2 p.m. Eastern. And here you can hear from Cell Maps for AI or CM4AI. And this is a data generation project which seeks to map the spatial temporal architecture of human cells and how you can use these maps towards the grand challenge of interpretable genotype-phenotype learning. There's going to be 5 presenters that will be involved in this presentation, so you can hear from their perspectives and all of the work that they're doing for the CM4AI program. Next, I just wanted to highlight a potential funding opportunity, and this is pilot projects enhancing utility and usage of common fund datasets. You can scan the QR code on the right here to get more information about this potential funding opportunity. But to give you a brief summary, there's many valuable and widely available data sets as part of the Common Fund programs. And we're hoping that this opportunity allows participants to demonstrate and enhance the utility of the selected data sets, including generating hypotheses and catalyzing discoveries. So as part of this, the applications closed June 23rd. And then as part of this, there'll be the 200,000 award maximum. And one last event that I just want to showcase, we have upcoming in 2 weeks is the Pink-based office hours. And this will be during the exact same time during our Knowledge Portal Network webinars, as you can see here. And so if you're already registered for this event, as you all are, you don't have to worry about registering for this one either. Here we'll be talking about identifying disease-associated cellular states in the pancreas. And there are several different topics that will be covered, specifically the definition of cellular states and gene programs in a cell type, approaches for determining which states and gene programs are involved in disease. And then we'll walk through some of the tools with a brief preview that's showcased on the right here. Again, if you have any questions or concerns about this office hour session, please feel free to let me know. And so with that, those are all of the different events which you can see from the Knowledge Portal Network. And so we're happy to have, you ask us any questions if you'll be participating at any of those. And so with that, we now have a presentation from Emily Griffin of the Gaulton Lab, who'll be showcasing her work on Tiger. So Emily, please feel free to take it away.
Thank you so much, Mackenzie. Let me see if I can share my screen here. Okay, wait one.
Looks great on our end.
Okay, looks good. Okay, great. Awesome. Okay, so as Mackenzie mentioned, my name is Emily Griffin. I'm a postdoctoral fellow in Kyle Gaulton's lab in the pediatrics department at UC San Diego. And today I'm excited to talk about our work, which we recently published in Nature Genetics on developing a machine learning-based model that can help us predict risk and identify heterogeneity in type 1 diabetes. So before we dive into our study, I wanted to talk a little bit about the background for type 1 and some of the motivation behind developing this risk score. So as many of you are probably aware, type 1 diabetes is a disease that affects over 2 million Americans, and it's characterized by the autoimmune destruction of insulin-producing pancreatic beta cells. While there are some environmental factors that influence the development of type 1 diabetes, we know there's a large genetic component, with heritability estimates being around 40% for monozygotic twin studies. And it's for this reason that the American Diabetes Association recommends screening for individuals that have an immediate family member with type 1 diabetes for the presence of autoantibodies. These autoantibodies are made early in disease during what we call stage 1, when the body begins to recognize beta cells as pathogenic. In stage 2, we see depletion of beta cells that results in hyperglycemia. And in stage 3, there's such extensive beta cell death that individuals require exogenous insulin administration to prevent complications of hyperglycemia. Now, I mentioned that autoantibody screening is done for individuals with an immediate family member who has type 1 diabetes. But what I didn't tell you is that only 1 in 20 of these individuals go on to develop type 1 diabetes themselves. And that means we're continually screening the other 19 out of 20 that will never develop disease. Perhaps more striking is the fact that 90% of people with type 1 diabetes have no family history at all. And as a result of a lack of global screening, nearly 40% of individuals are hospitalized with life-threatening complications of hyperglycemia at the time of diagnosis. So how can we identify who with a family history needs regular monitoring to prevent these life-threatening complications and who without a family history should be monitored at all? Genetics offers us a picture of a person's risk independent of their family history and where they are in disease development. So this means enabling monitoring for those high-risk individuals to prevent complications, but also it means providing an opportunity prior to disease onset for the use of disease-modifying therapies. So just a quick overview of our study. There are 3 main components that I'm going to be talking about today. First, we'll talk a bit about the work that went into identifying genetic risk factors associated with type 1 diabetes, which utilize genome-wide association studies. Next, we'll talk about how we built a machine learning model that uses these factors to predict a person's risk of type 1 diabetes. And then last, we'll go over some ways that we've been able to leverage information from this machine learning model to identify clinical heterogeneity in type 1 diabetes. So we can't talk about the genetics of type 1 diabetes without talking about the MHC, or the major histocompatibility complex. The MHC is a 4 megabase pair region on chromosome 6 that encodes about 50% of the risk for type 1 diabetes. The genes at this locus are densely populated with genetic variants, which form complex patterns of coheritability or linkage. And it's because of these complex patterns of linkage that it's been really difficult to resolve independent signals associated with disease. When we consider the rest of the genome, The other 50% or so of type 1 diabetes risk is spread out across more than 90 loci, where the effect sizes are much smaller compared to those in the MHC and require genetic information from very large cohorts in order to capture. So to uncover these independent genetic factors driving risk of type 1 diabetes, a graduate student in our lab, Carolyn McGrail, leveraged genome-wide association studies from over 20 cohorts encompassing 20,000 individuals with type 1 diabetes and 800,000 individuals without disease. Our meta-analysis of these studies identified 87 loci associated with type 1 diabetes, 8 of which were novel. And to resolve variants at these loci that were driving an association with type 1 diabetes, as opposed to those that were simply in linkage with the drivers of disease, we performed fine mapping on our 87 loci meeting genome-wide significance and an additional 10 loci identified in previous studies. Using SUSY FineMap, we uncovered 133 independent signals associated with risk of type 1 diabetes, and for each of these, we provide credible sets, shown here, which contain variants ranked by their probability of being the causal variant driving an association with type 1 diabetes at a given locus. Within the MHC, which I mentioned has complex patterns of linkage, we performed stepwise conditioning and identified 27 independent signals, many of which were previously thought to be dependent on linkage with known HLA alleles or amino acid polymorphisms. One example of this was a non-coding variant, shown here, which was strongly linked to the amino acid polymorphism HLA-DQB157, but it has its own independent effect on T1D risk. So these results suggest there are several independent signals within the MHC, which encode risk of type 1 diabetes that go beyond the traditional haplotypes associated with this disease. So now that we have our genetic factors, how can we use this information to create a model of T1D risk? So traditional genetic risk scores rely on fixed weights for each variant and multiply that weight by the number of risk alleles a person has. The sum of those weighted risk factors gives us a final score, which we can use to understand a person's risk of disease. However, this method falls short in two important ways. First, it fails to account for epistatic interactions between variants, which may influence the risk given variant imparts on the person. And second, it assumes that the risk of each variant follows a linear additive model, which doesn't take into account synergies in risk that may arise as allele dosage changes. So to adjust these limitations, we implemented a machine learning approach, which dynamically weighs independent risk factors based on the presence or absence of other risk factors. This allows us to utilize the entire landscape of genetic risk rather than considering these factors as fixed and in isolation. So to build this model, we used 129 lead variants from our independent signals found in our GWAS meta-analysis and added an additional 70 HLA variants that were previously identified in familial proband studies of type 1 diabetes. Very talented graduate student in Hannah Carter's lab at UCSD, TJ Sears, trained a classifier model to predict type 1 diabetes using 10,000 individuals with type 1 diabetes and 20,000 individuals without disease. He performed 10 cross-fold validations, which splits the data 10 times so that 90% of samples are used to train the model and 10% are used to validate the model. The output is a set of scores, which we can form T1D percentiles for so that future scores generated can be contextualized by where they fall in the T1D percentile. To evaluate the sensitivity and specificity of our model, we can take the scores from our cross-fold validation and generate a receiver operator curve to see how well our model classifies type 1 diabetes risk at a spectrum of thresholds. The area under this curve is a single value metric, which basically combines both sensitivity and specificity. So a number closer to 1 indicates perfect classification, while a number closer to 0.5 indicates the model performs about as well as chance. As you can see, our area under the curve is, or the AUC is 0.937, which significantly outperforms a previous linear additive risk score. And when we look at a cohort that's unseen by both of these models, this is the All of Us cohort run by the NIH, we see that our machine learning model shows even greater generalizability than the alternative linear model, likely due in part to the novel signals used, as well as the inclusion of these genome-wide variant interactions. So I mentioned that we can use machine learning to identify dynamic variant effects, but what does that look like? If we look at the impact of a handful of variants used in our model on the 30,000 people that the model was trained on, we see the impact of a given variant is not homogeneous across all individuals with a given genotype. So the genotypes here are split into mahogany, yellow, and this sort of light green. As we know, this reflects real biology because in biology, genetic variants don't act in isolation. And what this spread shows us is the influence of one variant heavily depends on the context of other variants an individual has. So what do these interactions look like? Well, we can pull out interactions between pairs of variants from the model, and we see that they exist both within the MHC, shown in purple, several of which have previously been captured, but they also exist between the MHC and the rest of the genome, as well as across non-MHC loci. One example of a strong interaction found in many individuals is between our novel MHC variant and the insulin variant, which highlights opportunities to explore how biological synergies between variants may be driving disease development. We can also quantify the impact of our model's interaction effects by splitting our cohort into 10 groups ranked by the complexity of their genetic risk. So individuals with low complexity, shown sort of towards the left here, have a low number of interactions and very few drivers of type 1 diabetes risk. We can see that our model performs here significantly better, but only marginally so relative to a linear additive model and a logistic regression that uses our same 199 variants as inputs. However, as we look at the individuals with more complex patterns of type 1 diabetes risk, they depend on an increasing number of interactions between risk variants. And we see that the model has more pronounced performance here relative to the linear additive scores, underscoring the importance of machine learning in enabling prediction of disease risk across individuals with diverse risk profiles. So last, I want to talk about some really exciting applications of the model in understanding both genetic and clinical heterogeneity. And I mentioned before, we can derive information about the impact of each variant on an individual's risk score from our model. This impact is what we call SHAP value, or Shapley additive explanation. We can use these SHAP values for all 199 variants across all 30,000 individuals used to develop our model to understand patterns of genetic risk. We do this by performing dimensionality reduction with PCA, which collapses all of this information into a 2D space, maximizing the difference between points. We then perform clustering using K nearest neighbors and select the number of clusters based on both inertia, which measures how close the points are to the centroid of each cluster, shown in red. And we also look at silhouette score, which measures how similar a given point is to the cluster it belongs to versus all other clusters. So using these metrics, we identified the optimal number of clusters to be 4, and we can then use these cluster assignments to investigate whether there may be significant biological or clinical trends between people that fall within these different groups. Because these groups are derived from SHAP values or variant impacts across all 199 variants, we wanted to first assess whether there were differences between groups at the individual variant level. And indeed, we see some pretty striking trends stand out here. Remember, I mentioned that the MHC comprises approximately 50% of risk of type 1 diabetes. What we see when we partition individuals into these 4 groups is that for people in this 4th cluster, we see a much larger impact of MHC variants than we see in the other clusters. And we know that this reflects real trends in individuals with type 1 diabetes because there's a subset of individuals who develop type 1 with little to no risk found in the MHC. So it looks like we're able to capture sort of this trend with our clusters. What's more interesting is that there are variants within the rest of the genome, such as at the signal PTPN22, which have long been associated with type 1 diabetes, but appear to have differential impacts on these different groups. And if we look at the picture overall, it seems like the individuals with a larger burden of MHC risk have a lower contribution to risk in the rest of the genome, while those individuals which experience smaller or more moderate risk from the MHC variants tend to have more risk spread out throughout the rest of the genome, so clusters 1 and 3 here. So how can we make sense of this from a biological standpoint, and how might the differential impacts of these variants be playing a role in disease etiology? We actually get a glimpse of cell types that may have diverging roles in disease when we look at cell type specific regulatory elements that are enriched for high impact variants in each cluster. So what we see is in cluster one, we have a large impact from variants that reside in regulatory elements of CD8 T cells, which we know are responsible for binding MHC class 1 receptors and initiating a cytokine-mediated cell death. Interestingly, this cluster 3 here has a large number of important variants that are sitting in regulatory elements of pancreatic acinar cells, which echoes some work done previously in our lab that identifies a possible role for these variants in type 1 diabetes etiology through their dysregulation of pancreatic enzymes. Now, if we look at the distribution of variant impacts across these different categories of annotations, so MHC in purple, T cell regulatory elements in yellow, pancreatic cell regulatory elements in teal, and innate immune cell regulatory elements in red, we see, as we sort of suspected, that risk in cluster 4 here is predominantly driven by variants in the MHC. And while these same variants have a lower effect in clusters 3 and cluster 1, these two clusters show higher impact of variants both within T cells and within pancreatic cells. So the work to understand some of the biological differences between these groups is ongoing in our lab, and we think it could highlight diversity of mechanisms driving disease development. So if these clusters are demonstrating differences in biological processes, an important question from a translational standpoint is, do we see distinct clinical differences between individuals in these different clusters? And before I show you the data, I just want to emphasize that, as many of you probably know, type 1 diabetes is not a monolith. There's a lot of heterogeneity when it comes to clinical presentation. So whether that's age of onset, the rate at which a person progresses through these early stages of disease, or associated comorbidities and autoimmune diseases. Type 1 diabetes, just like type 2 diabetes, does not look the same for everyone. And the field has been trying to find a way to understand these differences so that we can provide monitoring and treatment that matches both risk and experience of every individual. So we evaluated some of these clinical characteristics with the help of Alex Gavin, who's our pediatric endocrinology fellow. And I'm excited to show that the clusters of individuals do in fact demonstrate meaningful differences in clinical phenotypes, including age of onset, which is one of the most striking examples we found. So individuals in cluster 4 and cluster 2 seem to have a much earlier age of onset than those in clusters 3 and 1. When we look at rates of diabetes-associated complications, we see there are significant differences in instances of retinopathy, renal disease, cardiovascular disease, and neuropathy. And as you may know, a lot of these complications are behaviorally driven. That is, they are often associated with poor blood sugar management. But when we look at measures of HbA1c, which are a sort of proxy for average blood glucose over time, we see no significant difference between clusters, suggesting that the phenotypes measured here are really reflecting patterns of genetic risk over environmental contexts. So in summary, we've talked about this machine learning model Tigers, which predicts risk of type 1 diabetes by capturing dynamic impacts of variants on T1D risk and by leveraging genome-wide variant interactions. We've also seen that this model enables the discovery of both genetic and clinical disease heterogeneity. And as of April 30th, the model has made open access on Zenodo, thanks in large part to Parul Kudakar, who helped make running the model and streamlining it very easy to use for anyone. Some of the ongoing questions in our lab that we're addressing involve how can we do a better job of representing risk in individuals with diverse backgrounds. So we're exploring integrating multi-ancestry data to better capture risk in these underrepresented populations. We also want to evaluate additional biological and clinical characteristics of each cluster, and particularly since there's been a huge wave of disease-modifying therapies entering clinical trials for type 1 diabetes, we want to see if we can predict response to these therapies using either genetic features or cluster assignments themselves. And with that, I'd like to thank the Gaulton Lab, Kyle Gaulton, our PI, as well as Carolyn McGrail, Alex Gabin, and Parul Kudakar. Also the Carter Lab, who helped us develop the machine learning model, so led by Hannah Carter. We also had, as I mentioned, TJ Sears, who helped run the model, and fellows at the Broad Institute, Jason Flenick, and Patrick Smadbeck. And with that, I'm happy to take any questions.
Thank you, Emily. That was a very informative presentation, and I see we do have some questions in the chat. The first is from Nora Francini. How was the T1D defined in the AOU? And I believe this was in reference to slide 19 and 20 when you were presenting on there?
Yeah, the All of Us cohort. Yeah. So, Nora, that's an awesome question. It's been, as maybe you experience if you work in the All of Us data set, really difficult to identify who actually has type 1. So, for those of you who don't work with it, it's basically has a bunch of ICD-9 and ICD-10 codes, SNOMED codes. If you were just to filter someone based on whether they have an ICD-10 code for type 1 diabetes, you are going to get a lot of type 2 diabetes individuals just because of how these things are coded for insurance purposes. So we actually had to go in and look at very specific drugs that people were taking and other types of diseases or comorbidities they had in order to refine our understanding of who actually has type 1. So we looked at who was actually taking insulin for one. We wanted to rule out people that had gestational diabetes or steroid-induced diabetes, things like that. And so we have listed in our manuscript all the different criteria that we used in order to filter down to these individuals with type 1 diabetes, but we are open to hearing other thoughts people have. It's been quite a process in that biobank and in others to identify people with type 1.
And so I think someone put in the Q&A, and I think it's in response to this replication of the subtypes in independent biobank data using 199 variants. So I think that might be part of that as well, potentially.
Um, so I'm trying to find the question for that. Mm, I don't see the one you're referring to.
Sorry, it's in the Q&A.
Oh, I'm looking at the chat. I can't find the Q.
It was submitted as you were about to
Oh, I see.
explain, so I had thought potentially these
Oh,
interwoven.
yeah, I see. Yeah, so we were able to replicate this in the All of Us cohort. So we were able to project those individuals into the different clusters. We see about a similar distribution in terms of how many people are allocated to each cluster. And then we also were able to replicate some of the findings in terms of the complications associated with each cluster. So I think it was neuropathy that we saw most significantly, but we did have a much smaller cohort there. Instead of 30,000 people or 1,000 individuals with type 1, we only had about 300. So we lacked some power to identify all the same trends, but we did see some significant differences there as well.
Great. Thank you for that clarification. Um,
Mhm.
there's another comment in a question in the chat which asks, while genetic heritability or genetic predisposition do not explain all onset as explained in the background, how does this model using only genetic risk or nonlinear version can be accurate if the AUC is greater than 0.9?
Yes, that's a great question. I think so. Yeah, you're mentioning essentially that I said heritability was about zero point 0.4, and then we're able to really highly predict instances of type 1. I think that's a good question. I'm thinking when I think about heritability, I think about if one of your parents has it or your sibling has it, how likely are you to have it? And I guess if your twin has it, how likely are you to have it? And the answer is about 40%. We haven't done prospective studies with this score yet. So I think we have to implement this in the clinic and look and see how well it does predict in that instance. I would be curious to see if we're able to get a really high rate of prediction like we do for these cohorts. But remember, some of these cohorts already have like an underlying family risk because they're often recruited based on having a family history. So I think we're already enriching for people who have a high risk, and then you're laying genetics on top of that. So you're kind of like, to an extent, it's almost inflating how well it performs relative to how it might perform on a totally random group like the All of Us group, which it still does perform pretty well on. But I think to answer that question, how well it performs, I think we'd have to do prospective studies.
Great. Thank you for clarifying there.
Mhm.
Okay, the next question in the chat says, if clinicians want to use this model, do they need to generate their own PRS case and control distributions, or are existing distributions provided so clinicians can just run, say, patient VCFs and compare them?
Yeah, so a clinician would be able to. get a VCF, input it, and get both scores and percentiles relative to type 1 diabetes out. So we already have those percentiles. You don't need to get your own distribution. I will say that we're, as I mentioned, currently working on trying to get percentiles for different groups of different racial and ethnic backgrounds, which might be slightly different from like a Northern European background, which is what the data was trained on. So stay tuned for that. But what we do have right now is percentiles that we can give you based on like a European background.
Great, thank you. Um,
Mhm.
okay, there's another question which asks, are you also planning to use other clustering methods beyond k-means clustering? In type 2 DM, endotypes are classified using a Bayesian clustering method.
I think we hadn't really considered using a different clustering method. I think the difference between what we had done and some of the clusters for type 2 diabetes is instead of clustering based on allele frequency, is we clustered on the feature importance derived from the model, which I think is just a new sort of parameter that we were able to cluster on. Currently, we're seeing, all these trends in clinical phenotypes, as well as some of the work done by Alex Gavin in our lab has seen interesting differences biologically. So I think right now we feel pretty good about where these clusters have come from. I don't think we have plans to integrate a new clustering method, but that's a good suggestion.
Great, thanks. The next asks, do you validate the subclusters you identified in other independent cohort or in real patients with T1D or ABB plus?
Yes, so ABV plus is those autoantibody positive individuals. And yeah, so we used the all of us cluster, which was unseen by our model when we were developing it. But we're currently working with TrialNet to look at individuals with autoantibodies and different clinical features to see where they project in these clusters and what some of the features are that may distinguish these clusters that we don't currently have access to in the all of us data set or our original cohorts. And we're very open to any collaborations with anyone who has access to other independent cohorts that they want to look at in the context of these clusters.
Awesome. Um, so I'll give it another few minutes to see if there are any other questions that come in. But one thing that I think, we're interested in as part of the CMDKP is finding a way to integrate this into, our portal and things like that. So I think, that's something that we're hopeful that eventually, we'll be able to collaborate on that as well. And I know we haven't had a chance to fully discuss that. But I think that's something that, might be of interest to the community to help, expand this a little bit more. So,
Yeah.
yeah, I
No,
think
absolutely. I think Kyle's mentioned that as well, a collaboration there. And I think that would be really great. And we have, as I mentioned, we have someone working on further sort of characterizing these different groups or clusters or subtypes. And so we could provide some of that, hopefully, as like an interactive browser so people could take a look and see what are some of these different features. How do these SHAP values look across all individuals? As I mentioned, they're variable even within people that have the same genotype. So looking at kind of all of that information together might help people who are doing research understand sort of these different biological processes going on in these different groups and sort of the outcomes of those.
Yeah, I think like you said, having that interactive element is always, a great aspect of this, and being able to in real time see, how this information is, available and portrayed and things like that. So we're really eager to have that. I will give it another minute or so to see if there are any other questions. Just a reminder, if you want to ask your question, you can use the chat. Q&A, or you can raise your hand and we can take you off mute depending on what you're most comfortable with. But I know we already had a very lively conversation already, so I'll just give it, like I said, another few minutes to see if there's anything else that the participants would like to ask. Okay, there's one other question in the chat that's a follow-up to the validation of the subclusterings. And so for this, for cluster 2 and 4, they're very similar. So how do you understand this?
Yeah, so I can go back to this. So if we look here at clusters 2 and 4, cluster 4 is really driven predominantly by variants within the MHC. So these individuals probably are like They're not, we looked to see if they're partitioned by HLA haplotype. It seems like the DR3 and the DR4 haplotypes tend to really drive risk of type 1 diabetes in these individuals. But it's not that they're exclusively DR3, DR4, like compound hets or anything like that. However, in cluster 2, we do see that there is a pretty big level of importance for these MHC variants, but they also have risk within other variant groups that look like they're linked to pancreatic cell regulatory elements and T cells. So I think I would say that this is more, it's enriched for MHC variants driving disease risk, but they're not as important as they are for the individuals in this cluster 4. It looks like there's a bit more of like a broad trigger for type 1 diabetes risk in cluster 2 relative to cluster 4. And again, we're working right now, hopefully submitting in July or so on a paper that further characterizes each of these groups. So stay tuned for that.
Awesome. There's another question about the clustering. Can you show one more time how you cluster variants?
Yes. So we didn't cluster variants, we clustered people. Um, So each of these points is a person. So each person has 199 different values, one for each variant, that sort of tells you how important each variant in the model is to determining that person's risk of type 1 diabetes. So it's multidimensional data, and we performed PCA or principal component analysis to shrink this into a 2D sort of plot, which basically maximizes the difference between the different um, features. Let's see, what is the variant-based feature? So the variant-based feature is the SHAP value, which I mentioned earlier. It's essentially the importance, and it's kind of hard to explain maybe over here, but the SHAP value essentially tells you how important a given feature is for, or a variant is for determining risk of type 1 diabetes for an individual. So a variant with a high feature importance that's positive pushes an individual towards a likelihood of having type 1 diabetes, and a feature importance or SHAP value that is negative pushes an individual towards lower diabetes risk. So we can take those and we can cluster based on them using PCA, and then K nearest neighbors clustering, and then we can assign those different individuals to the different clusters based on where they sort of project. Okay, I see more. This person's got a lot of
Yes.
thoughts.
Yeah, no,
Feel
this is
free
great.
to.
Love the lively discussion.
I know, I know. Feel free to also email. I don't know if my contact information's anywhere, but did we consider partitioned PRS-based approach too? Partitioned PRS-based approach. So I guess what you're referring to is partitioning based on the risk score itself, like the different sort of centiles maybe a person falls in. Um, if that's the case, we think that I understand that approach for sure. I think this maybe is a bit more informative, because instead of just looking at whether someone's high risk or low risk, we're looking at more of the nuance of like what is the role of each variant in the context of all the other variants. So what is their overall landscape of genetic risk look like? And that's much more of a rich source of information than just kind of a single digit on a scale, right? So like you could have two people that are very high risk, but that risk may look very different for those two people based on what their genetic makeup is. And so I think by clustering based on these different SHAP values, we can get a better picture and better resolution of these different groups of individuals rather than just looking at high versus low risk. I don't know if that answered the question or not, but person's gone quiet, but maybe. Oh, yeah, you're
Thanks.
welcome.
Yep. Okay. That's perfect. Great. Any other questions? We do have some time to take a few more if there's anything else that comes up. And as Emily said, please feel free to reach out to either us or Emily to answer any more in-depth questions that you might have. But again, I'll wait a couple seconds to see if there's any others. But again, thank you all for this discussion that we've already been having. Okay, I'm not seeing any others at this time. So I think, if you have any questions beyond what we've discussed, please feel free to reach out. Thank you so much, Emily, for joining us today to walk through all of this. Like I said earlier, we're really excited to collaborate on, what we can do in the future with both this work and the portals. But again, because we've been talking so much about the All of Us data, I just want to one more time invite all of you to join us on Monday for the roundtable, because again, some of you might be interested in that after, this discussion as well. So just a reminder, this is going to be Monday from 1 to 2 p.m. Eastern. Please feel free to scan the QR code or use the link below, as we'll be talking about some other examples using the All of Us data. And just a reminder to everyone that we will be meeting again in 2 weeks, and we're very excited to have a discussion about pink base, as I'd mentioned before. So lots of events coming up that we can't wait to continue to talk to you all about all of this. So thank you again, Emily, for joining us, and thank you all for the participants for joining us at the webinar today. And we look forward to hearing from all of you and seeing you at our next sessions. So hope you all have a great rest of your day. Thank you again, everyone.
Thanks, Mackenzie.
Bye.